Hello, students and viewers. So, this is the second part of chapter number 14, that is structure and types of RNA. So, in first part, we have learned about mRNA. Now, this is the second part of this chapter. In this chapter, I am going to discuss in detail about rRNA, that is ribosomal RNA. Now, here you can see in each of these RNAs, you have to learn structure, its structure and functions, right? So first see structure of rRNA. So ribonucleo, ribonucleic acids is the type of RNA that is the part of ribosome, right? So rRNA is the part of whom? That is the part of ribosome. So it is defined as the molecular machine. See, ribosomal RNA is defined or described as what? Molecular machine. Why? Because that catalyzes the synthesis of proteins. Right? So we have different kind of amino acids. Total 21 amino acids are there. And from amino acids, again different proteins they are formed. Right? So the formation of protein is takes place because of this rRNA. So hence it is called as molecular machine, right? It makes up to 60% of the weight of ribosomes since they play major role in the function of ribosomes such as binding to mRNA, the recruitment of tRNA and catalyzation of polypeptide bond formation between amino acids. So these are the major functions. See, binding to mRNA, first job of rRNA, right? Second job, what it is a recruitment of tRNA. See, what it does the recruitment of tRNA. Why? Because tRNA carries particular amino acid, right? And then again it catalyzes the peptide bond formation between the amino acids. So these are the three main important role played by rRNA. Okay, please do remember binding to mRNA, recruitment of tRNA, and peptide bond formation between amino acids. So they also determines the shape of ribosomes using rRNA core. So rRNA core helps to determine the particular shape of ribosomes. So here we know that there are different kind of ribosomes. They are present in prokaryotic cells as well as eukaryotic cells. So earlier in this difference between the prokaryotic and eukaryotic organisms where we have learned, right? In case of ribosomes we have learned, or we used to say in case of prokaryotic it has the 70s type of ribosome. In case of eukaryotic it has 80s type of ribosomes, right? So now S is nothing but what? It is Swedberg unit. S stands for what? Swedberg, the scientist who discovered this Swedberg unit. So on this name S is given. Swedberg. These are the S means Swedberg unit, right? S. So now here see 70s. Suppose we uh, have this 70s type of this ribosome, then again it has this is somewhat larger you are observing. So this is the larger subunit of ribosome, whereas this is the smaller subunit. Means each ribosome has its larger subunit and this is smaller subunit, right? Larger and smaller. So 70s ribosome is again categorized or made up of this 50s subunit and 30s subunit. See, suppose you do the total of this 50 and 30, it comes 80, right? But we this is the again higher studies. In higher level, in case of your post graduation, you will go into the detail why this 50/30 is there and why only this is called as 70s type of ribosome. Okay, I will not go into the detail at first year here, but just remember 70s type is divided into 30s and 50s. So again 50s subunit, it has again see 23s ribosomal RNAs, then it again has 5s ribosomal RNA plus 34 different proteins. So this 50s is made up of all these 23s rRNA, 5s rRNA and 34 proteins. Again smaller subunit, it is again made up of 16s rRNA and 21 proteins. In case of eukaryotes, here again you have ATS type of ribosome. Always do remember in case of eukaryote, this ATS type is present. 70S found only in pro and ATS is found only in eukaryotic organisms, right? So 80 again divided into 40 plus 60, right? 60 is larger subunit and 40 is smaller subunit, right? So again 60 40 it's 100, but actually it's 80s, right? So again 60s is made up of 28s rRNA, 5s rRNA and plus minus 40 proteins, right? And again 40s subunit, it is made up of 18s rRNA and plus minus 25 proteins, right? So please do remember it has again different these smaller and larger subunit compositions in case of prokaryotic and eukaryotic. In case of prokaryotic, here you can see in a 30s it has 16s type of rRNA, but here in smaller subunit of eukaryote you will have 18s type of rRNA, right? Here you will observe 23s type of rRNA in case of prokaryotic larger subunit. Here in case of eukaryotic 20s type of rRNA is there in case of larger subunit, right? Rest of this 5s is common in case of both pro and eukaryotic cells, right? So likewise these are the ribosomes subunits which are present in pro and eukaryotic organisms. Now, the rRNA has distinctive 3D shape which is made up of internal loops. I will show you the image. Helices that creates specific sites that A, P and E within the ribosome. So this site also I will show you in my next coming slide. Just I will cover this. The P site, see total how many sites are there? 3 sites are there, right? A, P and E, right? So P site, this site functions as binding site for growing polypeptide, right? P site is act as the binding site for growing polypeptide, right? While A site acts as anchor to an incoming tRNA, which is charged with amino acids, right? A site, it is anchor site, we can say, or we can say this is the acceptor site for tRNA, right? So after the formation of peptide bond, the tRNA binds to E site briefly before leaving the ribosome. So we have here A site, P site and E site, right? E site where this tRNA is coming, then it will move towards P where the actual codon anticodon binds and polypeptide chain is elongates here in P site, right? And then it will move towards E site and then it will exit from this place, from the ribosomes, right? Then Additionally, the rRNA has the site for binding to the ribosomal proteins. It analyzes and distinguishes the residues of rRNA from protein. Yes, right? So, as it helps in the formation of protein, it analyzes as well as distinguishes the residues of rRNA, the residues of RNA, right? The proteins on the ribosome surface stabilize its structure by interacting with RNA core. Next, now here you can see, just take me this eraser. Okay, yes, you can observing here. This is the structure of a ribosomal subunit. You can see here, this is the larger subunit which is mentioned. This whole, right? I'm giving here outline for this. This is the larger subunit, right? Whatever I just made the outline with red color, and let me change the color of my pen. This is in purple color. I have made this. This is the smaller subunit, right? This is smaller subunit. See, so ribosome is made up of larger subunit and smaller subunit, right? Now this is the mRNA. See, again mRNA is there, which is present between this larger and smaller subunit. And then you are observing here, this is the A site, then this is the P site, and this is the E site. Now what is this? A stands for, P stands for, and E stands for. So see here on the upper side I have mentioned A site means what? Incoming amino acyl tRNA, right? This is incoming amino acyl tRNA, the tRNA which carries the amino acid, right? For amino acyl, the A site name is given, right? In case of P, now P is nothing but what? So now see, most recently amino acid, that is peptidyl tRNA, right? So this P site where the peptide chain is growing, and hence the name is given P site. Only remember, peptide chain is growing on P site, whereas E site means exit. Are you getting? I have mentioned here again red. A, A site. Then here it is P, P site, right? And then here it is E, which exits the this deacylated tRNA. Means amino acid is given, now it is empty, right? And hence it is called as what? The E site, right? So here also you can see this is the particular tRNA which is coming, which carries these amino acids with it. Then it arises on this A site, right? And then it will move to P site. Then whatever this anticodon which is there, okay, to the tRNA carries what? Anticodon. Then codon where it is present. So codons, they are present on mRNA. Please do remember whatever here you are observing. See here. See my pointer. Let me take pointer. Here you can observing this is G, U, G, C, A, C, A, A. So all these are what? These are the codons which are present on mRNA, right? Then anticodon. See my pointer. Anticodon are present here, right, at the base of this tRNA, right? So these anticodon and codon on the mRNA, they binds together, and then amino acid which is carried with it will be released, right? So it will be present, or it will be uh, I can say ready to join with the amino acid of another tRNA, right? So likewise this peptide chain will grow. Now this here in the P site it will means amino acid will be joined. Polypeptide chain is starts growing, and then this empty tRNA is moved from P site to E site, right? And then from the E site that will be thrown out of this ribosomal subunit, right? Now the tRNA function is over. The tRNA function is what? To carry the amino acid, and it's anchored to the particular A site which is present on this ribosomal subunit, and then it will interact with the codons of mRNA, right? Now, so this is the 3D weaves of ribosomes. So here you can see rRNA in dark blue, right? So this part, whatever the dark part you are observing, this dark, all this dark, so this is small subunit, right? And dark red, this is the dark red again, which is, see, this is the dark red, this is a larger subunit. The lighter color represent ribosomal proteins. So whatever the lighter color, let me change the color. I will take green. See, this is the lighter color you are observing, right? This is the lighter color. This is also a lighter color you are observing. So this lighter color, they represent the ribosomal proteins, right? Now, functions of rRNA. Now, so the rRNA is synthesized or transcribed in the cell nucleus. Yes, ribosomal RNA, they are synthesized and transcribed where? In the cell nucleus, specifically in the nucleoli, right? So in the nucleoli, this particular rRNA is synthesized. Please do remember the place where this rRNA is synthesized. The nucleoli play a major role in the biogenesis of ribosomes, right? Via the sequestration of ribosomal protein. Mm. Then come to the next function. Both pro and eukaryotic ribosomes are made of a large and smaller subunit. So just I discussed here. This is the larger one and this is the smaller one. Okay. So likewise, larger subunit and smaller subunit is present to the ribosomes of both pro and eukaryotic cells. Now, next, the small subunit of the prokaryotic are made up of an RNA molecule about 1500 nucleotides in length with Swedenborg coefficient of 16s. Right. So in case of prokaryotic, here it is given. It is made up of total 1500 of nucleotides. Right. So whatever the structure, see, let me change the color. Whatever this larger subunit you are observing as well as this smaller, so this subunit it is made up of what? Nucleotides. So whatever the nucleotide we are observing here in on the mRNA, right? All these codons which are present, right? So same like these nucleotides, they are present on this, means all these smaller and larger subunit it is made up of nucleotides only. So about 1500 nucleotides they are present, right, with Swedenborg coefficient of 16s. The small subunit along with the ribosomal protein has sedimentation rate of 30s, right? So it is this is the paired with larger subunit having two RNA molecules, one that is nearly 3000 nucleotides, that is made up of 23s type of ribosomes in length, and other in short sequence of 120 nucleotides, right? So these RNA molecules are accompanied by protein that gives rise to larger 50s subunit, right? So as we are seeing here 50 30, that means we are talking about which kind of ribosomes in from which organisms? 50-30, that is means this is the 70s type. So once you know the 70s, that means this is the ribosome of prokaryotic organisms, right? Now, the eukaryotic ribosomes are made up of two subunits, that is 60S. I have discussed again this earlier in my very second slide. 60S and 40S. 60S is larger subunit and 40S is smaller subunit. So smaller subunit made up of again two short rRNA molecule and that are less than 200 nucleotides in length. See, in case of prokaryotic, what we have seen, it is nearly 3000 nucleotides in length. See how much it longer, right? In case of eukaryote, it is less than 200. See, less than 200 nucleotides in length, right? So this is again small subunit is made up of 5s and 5.s type of rRNA, and the large subunit which is made up of two large molecules that are longer, one which has over 5 kilobase, that is 28s, and second will have the 18s, right? So 28s and 18s, it is present in a larger subunit of ribosome in case of whom? In case of eukaryotic organisms. Then again 28s, 18s and 5.8s molecules are produced by the processing of single primary transcript from the cluster of identical copies of single gene, right? So 5s molecule are produced from a different cluster of identical genes, right? Now see, totally eukaryotic ribosome has Wedberg coefficient of 80s, and in prokaryotic it is how much it is? 50. Sorry, 70s. Now, eukaryotic cells also have rRNA in mitochondria and chloroplast, right? So rRNA, it is also present in a mitochondria as well as in a chloroplast. Means eukaryotic cell, it has, see where it is present, in the nucleus and particularly in a nucleolus, right? So apart from this portion, it is also present where? In the mitochondria and chloroplast. You can see the structure, ultrastructure of these two, mitochondria and chloroplast. You will get the ribosomal RNAs structure. The ribosomes are associated with endoplasmic reticulum or can be free floating in the cell cytoplasm. So it is free as well as fixed. These are the two types of ribosomes are there. So free means what? Ribosomes which are free floating within the cytoplasm. That is free ribosomes. Whereas fixed means what? We have seen that the ribosomes, they are present on endoplasmic reticulum, right? This is the ribosomes which are present within endoplasmic reticulum, right? So these are the fixed type of ribosomes. So we also call it as what? R, rough ER. Rough endoplasmic reticulum means it has ribosomes on it. Whereas the one which is not present with the ribosome, we called it as what? Smooth endoplasmic reticulum means it is without ribosomes. So likewise, I have covered rRNA. So I think it is very much clear in case of pro and eukaryotic ribosomes and eukaryotic ribosomal rRNA. So thank you very much.